Okay, hi everyone. This is the lecture for chapter 2, which is our basic chemistry lecture, very basic chemistry. Obviously, most of you will be also taking general chemistry, but this is just a basic introduction. Very hard to separate biology and chemistry completely. Okay, so this first slide is just actually referring to the introductory little paragraph that's in the chapter. And it's about this rover named Curiosity that basically is on Mars and is assessing what types of elements are present there. Okay, so it says, did the planet Mars ever have conditions to support life? Right, so that's a question that you probably have heard of before. And so what this rover is doing, as I said, is essentially it has one part of it that can actually blast rocks, right? Laser blast the rocks to see if there's water there, because as you know, water is necessary to sustain life. And then there's another part of that rover called SAM, which it stands for Sample Analysis at Mars, and that uses spectrometry to basically look for or determine what types of elements are there, right? So particularly looking for carbon, hydrogen, oxygen, and nitrogen in the soil. Okay, so this, as I said, this chapter is focused on chemistry and it's just a very basic introduction. Okay, so we start off with the first part of the chapter, which is going to talk a little bit more about chemical elements, right? So I just mentioned a few in that introduction: carbon, right? Nitrogen, hydrogen, oxygen. Okay, and so let's talk about chemical elements a little bit more. All right, so some definitions: matter is defined as anything that has mass and occupies space. Again, some of these things are probably review, right? These first couple chapters, a lot of the material is review. And if it's not, then that's fine too. Okay, so matter exists in three states, right? Solid, liquid, and gas. All right. All matter, no matter what it is, whether it's living or it's non-living, is composed of elements. Okay. And so if we define what an element is, it's a substance that cannot be broken down into substances with different properties, right? So it's only composed of one type of atom. If we talk about what types of elements, right, make up the body weight of organisms, right, of living things, remember this is biology, the study of living things. There are 6 elements that make up 95% of that body weight of living organisms. And so if you want to think of an acronym to help you remember that, it's CHNOP, which what is that? Schnapps? I don't know. I'm not a big fan of acronyms, but if that helps, there's one there. You can probably make up your own acronym. If you wanted to. But so basically those 6 elements that make up 95% of the body weight of living things, of organisms: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Okay, and you should know these 6 elements. If we talk about sort of the difference or what types of elements are in the Earth's crust versus those elements that we just mentioned in well, not all of them, but in organisms, right? So remember, these are the 6 elements that make up 95% of the body weight of organisms, but there are also other elements as well, okay? So if you compare those, right, this is basically percent by weight, okay? In blue is Earth's crust, and then you can see the different elements here. Red is living organisms, okay? So you can compare what you got here. So for instance, silicon, right? Lots of it in the Earth's crust. You don't see any in living organisms, okay? Oxygen, both places to a pretty high percentage, okay? Nitrogen, carbon, hydrogen, these are all elements that are found in living organisms, but not in non-living matter, essentially in the Earth's crust. Okay, so I am not going to ask you to memorize anything in that graph, but if I showed you that graph, I could ask you questions about it. All right, so that's just meant to sort of be illustrative, just to compare the different elements that are present in the Earth's crust versus living organisms. Okay, so we have to talk a little bit about atoms, right? So atom is the smallest part of an element, okay, that displays the property of the element. And so it says an element and an atom share the same name, right? So we can say a carbon atom, okay? And those atoms are composed of particles, these subatomic particles, protons, neutrons, and electrons. Okay, so protons, neutrons, and electrons make up the atom. Okay, if you talk about the central nucleus of the atom, the center part, the central nucleus is composed of protons and neutrons. Okay, protons are positively charged, so P positive, protons are positively charged. This is just giving you an example of the weight, about 1 atomic mass unit. Let's pay attention to the charge here. Neutrons, no charge. Neutron, neutral, no charge, okay? Then around that central nucleus, you have orbiting clouds called electron shells, okay? So the electrons live in those orbiting clouds around the nucleus. They are not part of the central nucleus. Electrons are negatively charged. And obviously the charge of these particles is super important. Maybe not obviously, but the charge of these particles is super important in terms of how these atoms behave, right? Or how these elements behave in terms of what other elements do they make bonds with, right? Things like that. And we're going to talk a little bit about that in this chapter. Okay, so electrons are negatively charged. It says very low mass. So their mass is negligible, okay? Whereas protons and neutrons, each 1 atomic mass unit. For electrons, mass is negligible, okay? All right, so picture to just kind of illustrate some of these things that we just talked about. You can see the central nucleus of an atom composed of protons and neutrons. The electrons around the outside, orbiting around the outside, and they're there. Again, this is just summarizing what we just said on that previous slide, okay? All right. Now, let's talk a little bit further, right? Some other terms, and these are terms and things that you're gonna, find in the periodic table. Again, nobody's memorizing the periodic table. We're not really gonna even talk that much about the periodic table. That's something that you'll do a lot in chemistry, okay? But we'll mention it and talk a little bit about it. So each element is represented by 1 or 2 letters, right, to give it a unique atomic symbol. These are the symbols that you find in the periodic table. So for instance, H, hydrogen. We should be familiar with some of these main elements that we find in living organisms, right? Hydrogen particularly. And we're going to talk more about these molecules or these elements and then them making up molecules in our next chapter as well. Okay, so H stands for hydrogen, Na sodium, C carbon. Those are just some examples. Okay, so if we talk about the term atomic number, okay, atomic number is equal to the number of protons in each atom of an element. Okay, number of protons in each atom of an element. Mass number is equal to the sum of the number of protons and the number of neutrons. Okay, so atomic number, number of protons, mass number, the number of protons and neutrons. Okay, if you see the term atomic mass, it's essentially the same thing as mass number. So atomic mass, mass number is the sum of the number of protons and the number of neutrons. Atomic number is just the number of protons. Okay, again, we should know this. Oh, and again, reminder that we should have our exam 1 review sheet out right now, and we should be taking notes on it, right? Either electronically or handwritten, because we are going to submit that, and that does help you remember those things that we're talking about in the chapter. All right, so if we look at this symbol here, right, we see this atomic symbol, right? This is carbon. And again, this is how this will look in the periodic table. And so what do these numbers mean, right? So this number here, 12, that's up at the top left, is the mass number, and 6 is the atomic number. So again, remember, mass number is the number of protons and neutrons, okay? Atomic number is the number of protons. So by looking at this, if I asked you what is the number of neutrons, we should be able to figure that out, right? This is the number of protons plus neutrons. This is the number of protons. So 12 minus 6 will give you the number of neutrons. In this case, it happens to be the same, right? And it's the same as protons here, okay? You'll see that starts to change when we talk about isotopes and things like that. So anyway, mass number, atomic number. All right. Now, if we talk a little bit, like I said, we'll talk a little bit about the periodic table, but not too much. It's arranged obviously in a certain way. So atoms of an element are arranged horizontally by increasing atomic number, okay, in rows. These rows are called periods. Atoms of an element arranged in vertical columns are called groups, okay? And so any atoms that are within that same group share the same binding characteristics, okay? All the atoms that are shown in the periodic table are electrically neutral, okay? So it says the atomic number tells you both the number of electrons as well as the number of protons, because again, remember, atomic number is the number of protons. If this is neutral and these protons are positively charged, then we know it must have 6 electrons, which are negatively charged, right? So they will then essentially cancel each other out. And so that's what this is referring to. That's what this is referring to here. Okay? All right. So if we look at a picture here, again, this is just showing you a portion of the periodic table, pointing out, again, these are the, right, showing you the groups here, right? And then these are the periods here, these rows, okay? And again, you can see they're going by essentially ascending, right? Like we said here, increasing or ascending atomic number, all right? Again, to be honest, I don't even think I ever really ask a question about the periodic table on the exam, though now that I'm thinking about it, maybe I should, but I won't. So for the most part, we don't need to worry about focusing too much on the periodic table. But I mean, I guess if I showed you this and asked you, tell me what the atomic mass is of helium, right? We should know. Tell me what the atomic number is. We should know. Okay. And so basically, we should be able to read this. Okay. And so if we go back, right? Mass number, atomic number. Okay? All right. All right. So we can sort of move on from there. Like I said, we're not going to focus on the periodic table too much. Okay. So again, we said those elements that are in the periodic table are all neutral. Okay? Now, elements aren't always all neutral. Okay? So if we talk about isotopes, right? You can basically they're basically atoms of the same element, but they're going to differ in the number of neutrons, and then therefore they have different atomic masses. And so here, what this is showing you here is, right, that you have, this is carbon 12. This is what we just saw before. Okay. You have 6, so sorry, the atomic mass is 12, right? 6 protons, you have 6. Therefore, then you have 6 neutrons, right? Here, though, if you look at carbon 13, right, your atomic mass is 13. Your atomic number is 6, so you have 6 protons. And in this case, how many neutrons do you have? You have 7, okay? So when we talk about isotopes, they'll differ in the number of neutrons. Sorry, I said, I didn't mean to say when I said isotopes and said neutral, they're not neutral. That's not what I meant. I was referring to that they have a different number of neutrons, okay? So I apologize for sort of saying the wrong thing. Anyway, okay, so, and then if you look here, right, carbon 14, okay, it's called carbon 14 because its atomic mass is 14. Its atomic number is 6. Again, it has 6 protons, but how many neutrons does it have? 8. Okay, so I definitely can ask you a question where I show you something like this and I said, based on what you see here, tell me what the number of neutrons is. Tell me what the number of protons is, right? So we need to know, therefore we would need to know atomic number. We would need to know, what all of those terms that we just talked about before mean, right? What does it mean? What is atomic mass? What is mass number? What is atomic number? Okay, and again, remember this was we outlined all of that here. Okay, all right. So hopefully that's clear. Again, if you have questions about any of this, make sure you just ask me when we have our next meeting. All right, so the one thing about isotopes, though, it's not only just about that they have a different number of neutrons, okay? A lot of them will spontaneously decay, okay? And when they do that, they'll give off energy, okay, in the form of rays and subatomic particles. And this sort of decay or this release of energy can be helpful to us. It can also be harmful as well, right? When we think about radioactive isotopes, that they can, they can damage, right? They can be damaging. They can cause cancer, what have you. However, we also exploit them and we use them in science and medicine, okay? And so this is giving you an example of uses of low levels of radiation, right? Of radioactive isotopes. And so it's showing you some imaging techniques here, right? So if you look at panel A, okay, basically it's showing you a scan here of the thyroid gland, right? Sorry, scan of the thyroid gland, okay? And in this case, right, so basically you can radioactively label anything, right? So in this case, if you're, again, looking for, essentially, you want to just specifically focus on the thyroid, right? You can basically radioactively label iodine, which will be picked up by the thyroid because it's involved in producing thyroid hormone. And then you can basically image it and look at the activity levels of the thyroid glands. Okay, so it basically, this is a way of imaging, looking at that thyroid, but also a way of sort of assessing activity, right? So you can basically see if there are normal levels of activity or not, right? Again, using low levels of radiation, meaning small amount radiation tagged to iodine in this case. In B here, this is obviously showing you it's imaging the brain, okay? And this is something called a PET scan, okay, which stands for positron emission tomography, PET scan. We've probably heard of that before. And so if you look at the different colors, okay, the sort of most active areas are these red and green areas. The blue is the least active, low activity, okay, high activity here. And basically what you do with this is you radioactively label glucose. The patient, it's basically injected into the body, and that glucose, because the brain utilizes so much energy and requires such high levels of glucose, you're going to see most of that glucose is going to go to the brain, and then therefore, again, once you image it, you're able to assess activity. Okay, so you could pick up areas, say after a stroke, right, where there would be super low levels of activity because maybe there was damage in that area. All right, so these are two examples of uses of low levels of radiation. Okay, so again, I could ask you about, give me an example of somewhere in science and medicine where we use low levels of radiation. Okay, and so this is an example, or these are two examples of that. For high levels of radiation, okay, so these are high levels, meaning there's a lot of radiation there, a lot of radioactive isotopes. But if you look at B here, this is focused in one specific area. If you notice for the low levels, right, you were injecting that radioactively labeled glucose into your entire body. Okay, so if you did that with high levels, you're going to kill off cells, right? Here, this is radiation therapy, right, which is super focused. So this is obviously showing you somebody with, brain cancer. And so they're basically going to focus that beam into that specific area where the tumor is, okay? And these are high levels of radiation, however, they're specifically focused. Also, high levels of radiation can be used to help prevent spoilage in fruit, because it basically can kill off fungi and bacteria that might be on the fruit, because once they're there, they'll start degrading that fruit, okay? All right. So those were some examples of uses of radioactive isotopes, or uses of radiation, okay? All right, so let's continue on, and now we're going to really focus a little bit more on the electrons, okay? And then from there, focus on molecules and the formation of bonds between these elements, okay? So we already talked about the electrons. Remember, the electrons aren't in the central nucleus of the atom. They are around the outside, okay, in these shells. It says they're attracted to the positively charged nucleus, right? So because they're negatively charged, you have this sort of attraction that takes place, and so it says it takes energy to hold those electrons in place. All right, so there's basically energy within that atom between the central nucleus and the electrons in those shells around the outside. All right, so it says it takes energy to push them away and keep them in their own shell. Okay, The more distant the shell, the more energy it takes to hold those electrons in place, again, because they are drawn towards the center, okay? So they have energy, okay, due to where they are, because again, of this push, of this sort of attraction towards that positive charge. And we call this potential energy. All right. And so electrons are what's going to determine the chemical behavior of atoms. Like I said, what other elements they're going to form bonds with and what have you. Okay. And then therefore, if we're talking about forming bonds, we're talking about making molecules. All right. So it's really solely has to do with the electrons. Okay. How many electrons they have, where they are, and what have you. And so we're going to talk about this more. So there's something called a Bohr model, which is used to visualize electron location. Okay. And so, like I said, they revolve around the nucleus in these sort of energy shells, okay? And each of those shells has a particular number of electrons that they hold, all right? So it says for atoms with atomic numbers of 20 or less, the following rules apply. The first energy shell can hold up to 2 electrons, and then each additional shell can hold up to 8 electrons, okay? So basically the first shell, the shell that's closest to the nucleus is filled first, and then the ones further out are filled next, okay? All right, so again, this is kind of what we're going to follow because this covers most molecules that we're going to talk about, most elements that we're going to talk about in biology. All right, so that outermost energy shell is called a valence shell. All right, so we should know that terminology. And again, that valence shell is the most important because it's that outermost shell that's going to determine how that atom behaves chemically. Again, how does it form bonds? What does it form bonds with? Okay? Now, there's something called the octet rule, okay? It says that the outermost shell is most stable when it has 8 electrons. Therefore, atoms or elements are always trying to have 8 electrons in their outermost shell. And again, it's that's going to dictate how and when and who they bond with, okay? There is an exception to this. Of course, there's always an exception, right? If an atom only has 1 shell, that outermost valence shell is complete when it has 2 electrons. Because remember, if you remember what I told you back here, that first energy shell can only hold 2 electrons. Okay, so if you're talking about hydrogen, right? Its valence shell is complete when it has 2 electrons, not 8. Okay? All right. I already said this to you, okay? Just to put it in words there and I guess be a little more specific. The number of electrons in that valence shell determines whether the atom will give up electrons, whether it will accept electrons, or whether it will share electrons. Those are all basically different ways of forming bonds. All right? All right. I think we already kind of said this, right? Atoms that have their valence shell full are stable, okay? Atoms that do not have their valence shell filled are chemically reactive, okay? Because they want to have that 8, those 8 electrons in that valence shell. All right, so these are some Bohr models of atoms. And again, if I showed you one of these and asked you how many electrons are in the valence in its valence shell, okay, we would count the number of electrons in that outermost shell. 1, oopsie, that's not what I meant to do, right? So for instance, if we look at phosphorus here, 1, 2, 3, 4, 5. There are 5 electrons in its valence shell. And for our purposes in biology, we're really just paying attention to this outermost shell, okay? And so remember, how many electrons does it want to have, or what's going to give it stability is 8, right? So it's looking to fill that valence shell. Oxygen here, 1, 2, 3, 4, 5, 6, okay? 6 electrons. Hydrogen has 1 electron in its valence shell. And remember that this is an exception, okay? It's only going to take 2 to fill that, right? So again, we should understand what these are showing, and I could very well put pictures of these on the exam. I'm not going to ask you to draw these yourself or anything like that, but if you saw these, we should be able to determine how many valence electrons it has and basically what number it's trying to get to. Okay, so for carbon, how many? It has 4 electrons in its valence shell. All right. Okay, so moving on. So again, we're talking about the number of electrons in the valence shell. That's going to dictate how these elements behave chemically. Okay, and so now we can talk about the joining of these elements together to form molecules. Okay, so a molecule is two or more elements bonded together. And again, there's going to be different types of bonds that form. And again, this is all dependent on what's present in that valence shell. And we're going to see more of that in a second. A compound is a molecule that contains at least two different elements bonded together. Okay, so a molecule doesn't have to be different elements, compound, different elements. All right, so some examples of compounds, right? Carbon dioxide, water. Okay, we should be able to recognize these compounds. What's this one? C6H12O6. Think for a second. It's glucose, all right? A formula will tell you the number of each kind of atom in a molecule. All right. So again, here's that molecule that I just mentioned. That is glucose. Okay. So again, if I put up something like this, we should be able to understand what it is showing. Okay. This indicates there are 6 atoms of carbon, right? 12 atoms of hydrogen and 6 atoms of oxygen. All right. Okay. So like I said, now we have to talk about the different types of bonds, because if we're going to talk about forming molecules, right? Those are formed by bonding between elements. Okay, or between atoms, however you want to refer to it. All right, so it says bonds that exist between atoms in molecules contain energy. Okay, and again, those bonds are caused by interactions between those electrons in the valence shells, like we were just talking about. If we talk about the process of bond formation, forming a bond, that's a chemical reaction. Okay, all right, so different types of bonds. So first one we're going to talk about is ionic bonding, all right? And so ion is an atom that has lost or gained an electron. So think about that. If it loses or gains an electron, now it's going to have a charge, okay? It's no longer going to be neutral. And so in an ionic bond, electrons, right, are transferred from one atom to another atom. Okay, and the oppositely charged ions are attracted to one another. Okay, so an example of the formation of an ionic bonding is sodium chloride. Okay, and for the most part, we see, basically ionic bonds are formed in inorganic molecules, not in organic molecules. All right, so sodium chloride is an example. Sodium chloride, salt. So salts in general are solid substances that will usually separate, okay, in water and exist as individual ions. So when you put salt in water, it basically separates into sodium, a sodium ion, a positively charged sodium ion, and a negatively charged chloride ion, okay? So we'll see that. Let me just show you that right here, right? So here's your positively charged sodium ion and your negatively charged chloride ion. All right, so let's look at that kind of together. Just so anyway, here's your sodium ion, sodium atom, right? And you can see the sodium atom, how many, it only has 1 electron in its valence shell. Your chloride atom here has got 7. It's just looking for 1. So basically what's going to happen is this is going to move over here, okay? And so again, when that happens, right, you have sodium ion because it lost an electron, so now it has a positive charge. And then this chloride atom gained an electron, and now it has a negatively charged, okay? And then basically these join together to form sodium chloride because positive and negative are attracted towards one another, right? And so that's what this is showing you here. So again, example of an ionic bond, okay? Easiest example, sodium chloride. There are many other examples as well, but this is the example we're giving, okay? All right, another type of bonds, and these are, again, like I said, for ionic bonds, those are mostly, that's going to occur in salts, right, in inorganic substances. These other types of bonds we'll see in organic molecules, okay? And really, we're going to be moving forward through the course. We're focusing on organic molecules because those are the molecules that you find in living things. And so that's why I'm sort of drawing your attention to that. Okay, so covalent bonds, what are they? They result when 2 atoms share electrons, okay? So then each atom has that 8, that number of 8 in its outer shell, right? Again, remember that exception, that hydrogen, that outer shell is complete when it has 2 electrons, okay? Now, different types of covalent bonds. So a non-polar covalent bond, okay? Electrons are shared equally between atoms. So examples of non-polar covalent bonds are in hydrogen gas, oxygen gas, and methane. I'll show you a picture in a second. Okay, so that's what this is showing you here. So there's an equal sharing of these electrons. And so if you look at the structural formula, this is it's just straight, right? Everything's sort of equal and sort of, what's the word I'm looking for? Shoot, I can't think of the word. Anyway, they're shared equally, okay? And you'll see the difference in a second when we talk about polar covalent bonds, okay? So in polar covalent bonds, those electrons are not shared equally. They are shared unequally. The biggest example of a non-polar covalent bonds is in water, okay? And the reason why it's super important to talk about this is that when you have these when you have polar covalent bonds form, what ends up happening is you generate something called like a partial charge, okay? And so this is what this is referring to, electronegativity, okay? The ability of an atom to attract electrons towards itself in a chemical bond, okay? So in water, and I'm going to show you a picture and it'll make more sense in a second, the oxygen atom is more electronegative than the hydrogen atoms, okay? And the bonds are polar, okay? So just like we saw before with those other examples I gave you, say of like the hydrogen gas, where you just saw those 2 hydrogens exactly in plane with each other, that's not the case here in water, okay? So, you know what I can do? No, I'm not going to be able to do that. So if we were in the classroom, I probably would go onto the whiteboard, and I can clarify this via Zoom with the whiteboard on Zoom. But bottom line is, you know, if this was non-polar, right, and things were shared equally, you would see these hydrogens up here and be exactly in line with those oxygens, okay? But that's not the case. So what we just said is that the oxygen is partially negative. The hydrogens are partially positive. So that means they're going to attract negatively charged compounds, right? And so again, that's why you have this unequal sharing, and that's why we say these bonds are polar, okay? And this molecule itself is polar, okay? Because of this unequal sharing, okay? So again, it's not an ion. It's not a complete negative charge, but the oxygen has a partial negative charge, and you denote it this way. The hydrogens have a partial positive charge. You should know this, okay? All right. Now, because of water, right, containing these polar bonds, okay, and having the partial negative and the partial positive charge, it has properties that you don't see in other molecules, properties that are super, super important to sustaining life, okay? So the next part of the chapter focuses on water and talking about those properties, all right? So a little bit more about that. So again, we said water is a polar molecule. Again, what does that mean? Contains polar bonds, polar covalent bonds. What are polar covalent bonds? That's when you have an unequal sharing of electrons, right? And this is what occurs versus equal sharing, where there is no partial charge that's generated. Therefore, this molecule wouldn't be attracting any other molecules with a partial charge or ions or anything like that, okay? But with water, because you have the partial negative and the partial positive charge, you're going to see this is going to dictate how it behaves chemically, okay? All right. So here's another bond that's thrown into the mix here, okay? That's particularly, we talk about it mostly with water, all right? But not only with water, but a lot. If we're going to talk about hydrogen bonding, you got to talk about water. Okay, so again, because of the polarity, because of the partial negative and the partial positive charge that we talked about, hydrogen bonding is possible. Okay, so really any polar molecule, you would see this. All right. So what is a hydrogen bond? It's a weak interaction between a slightly positive hydrogen atom and a slightly negative atom. Any other slightly negative atom. It could be another, an oxygen atom and another water molecule. But basically, a hydrogen bond is an interaction between a slightly positive hydrogen and a slightly negative atom. Okay? It can, like I just said, it can occur between atoms of different molecules or within the same molecule. And basically a single hydrogen bond is not very strong, okay? It can easily be broken. However, multiple hydrogen bonds collectively together are very strong, okay? Hydrogen bonds are involved in maintaining proper structure of DNA. There are hydrogen bonds that form between the bases of DNA, right? So we talk about like A, T, and G, C pairing with each other. That's hydrogen bonds that join them together, okay? So there are, like I said, we're talking about hydrogen bonds in reference to water, but there's other molecules where you see this as well. All right, so just to kind of go a little further with this, right? Again, if we're looking at a model of water, right? We see it here. It's unequal sharing. Okay, you can see though it's sharing, but it's unequal. Okay, and it's because of that you have this partial negative charge of oxygen and partial positive charges of the hydrogen. So if you put a whole bunch of water molecules together, right, you're gonna have the partial positive charge of this hydrogen attracting the partial negative charge of this oxygen, right? And so that's all happening sort of collectively with all of these water molecules. And so that's what holds them together, okay? All right, so again, that dictates these properties of water that we're going to talk about, right? So I just said that water molecules cling together because of this hydrogen bonding, okay? And again, this is what's going to dictate its special properties. It says, without hydrogen bonding, water would freeze at minus 100 degrees Celsius and boil at minus 91 degrees Celsius, okay? That's not going to work on Earth, right? No way that water or life would be able to be present on Earth if that was the case, okay? So hydrogen bonding, just what we just talked about alone, basically allows water to be a liquid at temperature that's normally found in the Earth's crust, okay? So let's talk about some of these properties. And so these properties are listed in red. So the first one, water has a high heat capacity. And again, pretty much all of these properties that we're going to talk about are dictated by the formation of these hydrogen bonds, okay? So it says the presence of many hydrogen bonds allows water to absorb a large amount of heat without a great change in temperature. So when we say it has a high heat capacity, it means, again, it can absorb a whole bunch of thermal heat, but itself will not change temperature a lot. So that means if you think about environments or climate at the coast, right? Coastal climates are cooler because of that presence of that water and its ability, this high heat capacity that it has, okay? So it says a calorie is the amount of heat energy needed to raise the temperature of 1 gram of water 1 degree Celsius. Now, other molecules do not have a heat capacity even close to water, okay? So again, that's why water is essentially that molecule that sustains life, okay? All right. So again, if you think about climate on the coast, that's one sort of thing to think about related to this. Also, if we talk about maintaining body temperature, right? Maintaining internal temperature, lots of water in your body, right? Lots of water in your cells. That's what allows that to happen, right? And so the water provides protection from rapid temperature changes, which is obviously pretty serious if you talk about sustaining function of your cells, okay? And of your organs, essentially. All right. So what this is basically showing you is, let me just see something. We have the, oh yeah, okay. So this kind of goes along with this high heat of vaporization. So I'll go back to that in a second. It says hydrogen bonds must be broken to evaporate water, okay? And so bodies of organisms cool when their heat is used to evaporate that water. So again, it takes a lot it takes input of energy basically to vaporize or evaporate water. Okay. And so that is used, that property is basically used or exploited when we think about cooling mechanism. Okay. And so that's what this is showing you here. So, I mean, even if you just think about sweating, right? If that sweat produces water on your skin, right? And when that water is changed from a liquid to a gas, okay, it requires heat energy to go in to do that. And so that heat comes from your body, and that's what cools you off, right? And so that's what this is showing you, sort of how many calories or how much energy is required to change water from a liquid to a gas. Okay, and so this is showing you the evaporation part. This is showing you, if we're talking about, right, it's a solid here, freezing occurs here, and then moving, how many calories moving from a solid to a liquid. Okay, we can see a lot of calories or a lot of energy input required to evaporate that water. And again, that's why this process is super important in cooling. Okay, so like I said, this goes along with that next property of water, this high heat of vaporization. All right, another property, water is a solvent. Okay, and so what's a solvent? It's something that basically will dissolve solutes, right? Solids. And so it's a good solvent because of its polarity, right? Remember we said it has this partial positive and this partial negative charge. And so any other molecules that also have a charge or a partial charge, they'll be attracted to that water. And that's essentially how they dissolve and why water is a good solvent, okay? So some terms we hopefully maybe we've heard of before, but we need to be familiar with. So hydrophilic molecules will dissolve in water. So hydrophilic molecules, generally other polar molecules, right? Other molecules that, or charged molecules, right? Other molecules that have a partial charge, okay, will dissolve in water. Hydrophobic molecules do not dissolve in water. So these are things like lipids that are in generally non-polar molecules, okay? All right, so that's what this is showing you here. Water is a solvent. We go back to our sodium chloride example, right? We said in water, you know, sodium chloride, basically it dissociates into sodium ions and chloride ions. And why? Because that sodium ion is positively charged, right? And so those negatively or partially negative charges on the oxygen will be attracted towards the positive sodium ions, and vice versa here with the chloride ions and the hydrogen, okay? And that basically the water molecules surround these ions, and that's dissolving it and acting as a solvent. Okay, another property of water. Water molecules are cohesive and adhesive. Okay, so what do those two terms mean? Cohesion, right, is the ability of water molecules to cling to each other due to hydrogen bonding. Remember we just said, I mean, we said this in the beginning. All of those water molecules have partial positive and partial negative charges. They then are all attracted to one another, okay? So for example, and we all know this, water flows freely, right? Water molecules do not separate. Why? Because of this cohesion, okay? If you think about surface tension, right? Due to cohesion, the ability of these water molecules to cling to each other and be attracted towards one another. Now, adhesion is the ability of water molecules to cling to other polar surfaces. Okay, so cohesion, they cling to each other. Adhesion, they cling to other polar surfaces. Okay, and again, it's due to water's polarity. So it mentions here something called capillary action. And so even if, I mean, there's a couple examples of this. We're going to talk about, I'm going to give you an example about water transport in plants in a second, but even if you think about transport in blood vessels, right? Blood's made up of mostly water. That water can basically adhere, right, to the sides of the blood vessels, and that helps it move. You know, there are these glass capillary tubes, these little skinny, tiny little tubes that basically, you know, we say capillary action. If you put a little spot, you pricked your finger, right, and a little spot of blood. If you put these little tiny little tubes on these hematocrit tubes, right, it basically will collect that blood by capillary action. It'll move up that tube, and then you can actually spin that, and you can get a percentage of white blood cells versus red blood cells. But anyway, that's not the only example of capillary action, but the idea is that these water molecules adhere to the surface of whatever they're in, and that allows it to sort of climb up. Okay, so I'll talk to you about that example in plants. So it says the stronger the force between molecules in a liquid, the greater the surface tension. Okay, so anything, if you think about surface tension in general, you see it with water, but any liquid, right, that there's a strong force between the molecules will have a large amount of surface tension. Okay, so here, this is what I was referring to. So if we think about trees, right, where's water going to enter the tree? It's at the roots. Okay, that's how plants take in water, from the roots. And I'm sure we all agree that the tree needs to get water all the way up here, right? And so how does it do it? It basically, there are vessels in the plant, right? That basically pull that water up from the roots. And because that water, one, has cohesion, the water molecules stick to one another, adhesion sticks to the side of that vessel, right? It's able to transport that water up. If water wasn't polar and didn't have those properties of cohesion and adhesion, this wouldn't be possible, okay? And so that's what this is telling you here. So this is an example. If I asked you to give me an example of cohesion and adhesion, or an example of why cohesion and adhesion of water molecules is important, this is one example. Also, if you just think about transport of blood through your blood vessels, another example as well. Okay, another property of water, right? Ice is less dense than liquid water, okay? It's less dense than liquid water. So again, if we think about ice, if we think about a pond that freezes over, ice floats on top, right? Ice will be on top, and there may be liquid water at the bottom. Okay, so sorry, I said that and then I sort of skipped over the first two bullet points here, but it says water expands as it reaches 0 degrees Celsius and freezes. Okay, so you kind of see this, the water molecules, it says they become more rigid, but also more open. And so that's why you have this expansion, okay? All right. So again, like I said, ice is less dense. It floats on liquid water. Why is this super important? It acts as an insulator, right? So we're thinking about in the winter, there's fish, there's other aquatic life that's in a pond. If ice was more dense, it could freeze from the bottom up. It would just kill everything that was in that pond, right? But instead, because ice floats on water, it actually acts as an insulator and keeps that water temperature even warmer, okay? And so this is actually showing you here, obviously some seals sitting on an iceberg, right? Because it can float on the water. And so this is just showing you the density, okay? So less dense at ice, right? Compared to liquid water. All right, and so this is what I just showed you here. We think, or what I just showed you, what I just mentioned before. If we talk about an ice layer, this ice layer will actually insulate this pond. Again, if water was more dense, or ice was more dense than water, the freezing would happen here and basically freeze up. That's not going to work out. Again, if it froze here, it would just push all of that aquatic life up and basically cause it all to die. So again, I could ask you a question like that. Explain to me, why it's important that water or that ice is less dense than water. What would happen if ice was more dense than water if we in a pond in the winter, right? These are things to kind of think about. Okay, the last part of this chapter, super quick, we're almost done, is acids and bases, okay? So pH is a measure of hydrogen ion concentration in a solution. Okay, so when we talk about something being an acid versus a base, we'll talk about its pH. When water ionizes or dissociates, it will release an equal number of hydrogen ions and hydroxide ions, okay? And so acids, when they dissociate in water, will release hydrogen ions, right? Bases, when they dissociate, bases will either take up hydrogen ions, therefore decreasing the amount of hydrogen ions, or they'll actually release hydroxide ions. So a base is anything that has more hydroxide ions, right? And an acid has more hydrogen ions. And so when we talk about the measure of pH, it's the measure of hydrogen ion concentration in a solution, right? So let's talk about the pH scale. And again, we may have heard of this before, but basically it's a scale from 0 to 14. Anything from 0 to just less than 7 is considered acidic. 7 is neutral. Anything greater than 7 to 14 is basic. Okay, now the pH scale is a logarithmic scale. Okay, so each unit of change, meaning going from, say, pH 7 to pH 8, represents a 10-fold change in hydrogen ion concentration. So, right, it's not just a 1-fold change in hydrogen ion concentration, it's a 10-fold change. So here's another example, right? pH of 4 is 10 times as acidic as a pH of 5. Okay, but a pH of 10 is 100 times more basic than a pH of 8 because it's two values on that pH scale, right? So important to understand that. Again, when we talk about pH, it is really just the measure of hydrogen ion concentration. We talk about the differences between acids and bases, right? Acids have more hydrogen ions. We talk about bases, they have more hydroxide ions, but also bases can basically can take up hydrogen ions, okay? And so therefore they'd be decreasing the amount of hydrogen ions, right? So pH scale only measures the number of hydrogen ions. All right, and so this is and I may kind of, when I do the lab demonstration, maybe I think I might have some pH paper that I took home with me. Maybe we'll just pH a few common household items just to kind of look at this, right? So showing you a scale here, right? 0 to 7 here, acidic, hydrochloric acid, right? 0. Stomach acid, around 2. Lemon juice, around 2. Okay, tomatoes, 4. Coffee, around 5. Okay, so you can kind of get an idea here. Things that are basic, right? Obviously, sodium hydroxide on the very end here, right? So hydrochloric acid is just above 0. Sodium hydroxide, right? Towards the high end, right? Almost at 14. If we talk about egg whites, right? About 8, pH of 8. Ammonia, about a pH of 11. All right. Okay, one last thing that we need to talk about is something called a buffer, okay? And so what is a buffer and why do we care about it in biology, right? So a buffer is a chemical or a combination of chemicals that basically keeps pH within normal limits, right? Super important that the pH of your blood is maintained at pH 7.4, okay? If it deviates, either becomes more basic or more acidic, even slightly, it is life-threatening. Okay, so it's telling you this here. Blood pH drops below 7, acidosis results. Blood pH rises above 7.8, it's something called alkalosis. And this will lead to organ failure. Okay, they're life-threatening situations. So I hope you sort of, just by this, understand how important it is to maintain proper pH. Okay, so when we talk about a buffer, basically a buffer is going to say, so if we think about the human blood, right? The human blood has a really extensive and elaborate buffer system, okay? And so there are buffers there that maintain that pH to 7.4. So if that buffer system or those buffers weren't present, your body wouldn't be able to maintain that pH at 7.4. I mean, it would be almost impossible to do that, okay? So even with the buffer system in place, you still can have deviations of pH, if something major is going on, but that buffer system is there to maintain that pH. Without that buffer system, there'd be no way we would be able to maintain that pH. Okay. And so that's kind of what I just was referring to. It says the body has built-in mechanisms to prevent pH changes. Obviously, explaining the elaborate buffer system that exists within the blood is beyond the scope of this course, but just want you guys to be aware that it exists, okay? And we should know that human blood is around pH 7.4, right? Slightly basic, but obviously, pretty close to neutral, okay? But, and that it's super, super important that pH be maintained, okay? pH, again, if changes in blood pH occur, it is a life-threatening situation. All right, guys, thank you. That is it for chapter 2. Make sure you completed your review sheet as you are going through this chapter. If you haven't, then you can always sort of rewind and replay. All right, thanks.